Hello everyone, welcome back. Today I'll be covering chapter 10. I won't cover everything from chapter 10, just section 10.1, 10.2, 10.3, 10.4, and 10.7. So make sure you read those sections of the chapters. Let's do a quick review. We learned last week that proteins are encoded in DNA. And so all of the genes that our DNA is made up of serves as a code to make some type of protein. And so there are many different types. And so we go from DNA, the genetic code, to the copy of the genetic code, which is mRNA. mRNA is then spliced by enzymes, and so the introns are removed, and now it's ready to be translated into a protein. And translation occurs at the ribosome. Ribosomes, or the tRNAs, basically recognize the codons from the mRNA and translate that into an amino acid. The amino acids then are bonded together by peptide bonds. And so these are the amino acids. These are the peptide bonds fusing the amino acids together to form a polypeptide. And from a polypeptide, we also learned that folding of the peptide occurs in several steps. So it goes from secondary to tertiary to potentially quaternary, depending on what type of protein it is. And so we go from alpha helixes and beta sheets to different types of folds, depending on what types of charges are present on the protein. And then it becomes a fully mature protein unless it needs more processing. And if it needs more processing, then it goes to the endoplasmic reticulum and then out the door through the Golgi apparatus. All right. And so why do we care so much about proteins? Well, we learned that proteins are the essential parts of the cell membrane system and the internal skeleton. They are the glue that holds cells and tissues together. Proteins carry out biochemical reactions, many different types of functions. So they destroy invading microorganisms. So they work as part of the immune system. They act as hormones, as receptors, as transport molecules, and also as enzymes. Okay? And it's a very important feature that they have, and we're going to learn a little bit more about enzymes in a few slides from now. And so proteins are so important that they're even present in the replication of DNA, and the expression of genes depend on the action of proteins. So there are many different functions of proteins that are matched by their enormous structural diversity. Proteins are the link between genes and the phenotype. And so there's a direct link between a person's genotype, the proteins a person makes, and that person's phenotype. So for example, there's proteins that are function for eye color, and the phenotype is the actual eye color. So you have a blue phenotype here and a brown phenotype here, which is a set of observable characteristics. And so phenotypes, which is our physical characteristics, can be altered with genetic mutations. And so by definition, a mutation is a heritable change in the DNA sequence, meaning that you can inherit this mutation. But in general, it's a change in the DNA sequence. And so these can happen in any of your normal cells in your body and also to sperm and egg. Mutated genes can produce either an abnormal type of protein. It can produce a non-functional protein, so a protein that just doesn't work at all. A partially functional, meaning that it can slightly work, but not till its full capacity. Or in some cases, the mutation can cause no protein to be made at all. And so it can just completely mess up the code, and that means that the gene isn't even translated. Now, in some other cases as well, which I didn't include here, you can also have something that's called a gain of function, meaning that the mutation can actually cause something to be even more favorable. It can make that protein even more efficient. So there's a variety of things that happen with genetic mutations. It's not always all bad. And some other type of mutations can even affect just the timing and level of gene expression. So either there's just less being expressed or even more being expressed. While some others may result in no phenotypic change and pretty much are invisible. So you can there's probably a lot of mutations that we all have, except that those mutations aren't necessarily detrimental to us. That's why we're still alive. The detrimental types of mutations normally are lethal, and so you don't see them as frequent in a population. Mutations can also alter the amino acid sequence of a protein. So changes in proteins can produce mutations and change the phenotype, okay, from insignificant to lethal. So it just depends on what type of mutation it is. And so we'll talk about some examples in a minute. As we said earlier, not all mutations are harmful. There's about 400 different variants, different mutations in just hemoglobin alone. And hemoglobin is a very important molecule found on red blood cells, and it's a transporter of oxygen. And so this table shows here all of the different types of mutations that exist in hemoglobin. And it's just a single amino acid replacement. And so it's just one mutation. For example, this mutation is on position 6, and this one's on 7, 58, and whatnot. And so you'll notice that some of the mutations don't cause any harm at all to the whole protein alone. So it's still, even though this mutation exists, the protein works just fine. And then there's other mutations that cause detrimental effects. For example, a mutation on position number 6 with a switch to valine causes a disease called sickle cell anemia. And so you have another at the same position, which is a lysine, which causes hemoglobin C disease. Now, the position of the mutation does matter because it has to do with the protein folding. And so if the mutation is just in the right sweet spot to cause the protein to fold in a very abnormal way, then it'll lose its function. And so it looks like amino acid position number 6 might be a sweet spot. Whereas position number 7, although there's a mutation there, it doesn't necessarily change the structure of the entire protein. And so that's why all of these mutations, even though they exist, you still have a normal functioning protein. Let's talk about several different types of mutations. For example, a mutation that alters an enzyme. And first, let's define what an enzyme is, and then we'll talk about enzyme mutation. And so an enzyme, by definition, is a molecule that catalyzes biochemical reactions and converts the substrate molecules into products. And not only does it catalyze the chemical reaction, it actually speeds up the reaction. Meaning that if there was no enzymes, molecules, if you just leave it up to the environmental physics to break down a molecule, or if you leave it up to the environmental physics to build up a molecule, that would take years to centuries. And so enzymes are actually really neat because what they do is it's kind of like manipulating physics, manipulating energy. And another neat thing about enzymes is that they don't get used up in the reaction and they don't require energy. And so an enzyme is a type of protein. It's involved in biochemical reactions. For example, when you eat and you break down those food molecules, well, a lot of biochemical reactions are going on then. And importantly, and I'm repeating myself, it sums up all of the biochemical reactions going on in a cell. And so that is referred to as your metabolism. So if somebody says, what is metabolism? It literally is all of the biochemical reactions in which enzymes carry out. And so just take a quick look at this picture here. You should become familiar with the terms substrate and product. And so an enzyme, every enzyme will recognize a very specific type of protein or a very close variant, and that's its substrate. And so in enzymes and substrates fit like a lock and key. And so once an enzyme recognizes a substrate, it either will break it down or build it back up, just depending on what type of enzyme it is, and it'll create a whole different product. And so the molecule that it started off with ends up being a whole different type of product. Like, for example, when you drink milk, you're drinking a molecule called lactase. If you have the enzyme called lactose, then now you have broken down that that lactose in milk into glucose and galactose by an enzyme. And so enzymes take substrates and convert them into products. Enzymes also tend to work in like a chain of reactions. And so you have some enzymes that work alone. They don't need to work in a chain of reactions. And then there are other enzymes that work in a long chain of reactions. So for example, this figure here shows that a normal condition without a mutation. And so here you have gene A, which encodes for an enzyme. And enzyme A then takes a substrate and converts that into a product, okay? And now that product, it serves as a substrate for the next enzyme, which then breaks it down and then converts it into another type of product. And then the enzyme C, which is part of the reaction, this is the very last part, will take that, the product from the previous, convert that into a substrate because now that's enzyme C's substrate, and then break it down and then turn it into another type of product. But if you notice, this is like a step by step enzymatic reaction. And a lot of reactions within your cells happen this way. Like, for example, the breaking down of glucose to make ATP in the mitochondria, that requires a whole bunch of enzymes working together in a series just to break it down to make ATP. And if there's a mutation in an enzyme, and let's say that this enzyme is involved in an important metabolic reaction, so let's pretend like this metabolic reaction here is the breakdown of an amino acid into or the breakdown of a protein into some important amino acid or something. Then here you would start off with the protein. It'll be broken down by enzyme A, and now you split that protein in half, and those compounds that are made from that now serve as a substrate for enzyme B. However, enzyme B has a mutation, and now it can't do its job. And so compound 2 sits around. You're not going to end up with a product that the cells need. In fact, the product that you end up with here at compound 2 is not even useful to the cell, and that can be very detrimental, and that's actually what leads to disease. What leads to the disease is basically these proteins that have no function, and we talked about that in the last lecture too. So it's kind of like having garbage in the house. Garbage doesn't serve a function. You need to get rid of it, but sometimes your cells just don't know how to get rid of it, and so it can accumulate, and it can cause problems, which lead to disease. Here's a real example of a genetic disease, which is an enzyme that lost its function. And this disease is covered in your chapter 10, and it's called phenylketonuria, abbreviated for PKU, and it's a metabolic disorder. And so the enzyme that is responsible for breaking down phenylalanine is dysfunctional. And it ends up with this byproduct, which is called phenylpyruvic acid. And that's what gives the foul odor, which is associated with this type of disease. And so that's how it was first discovered. There is according to the textbook, a mother noticed that both of her children had very foul-smelling type of urine, and so she took the kids to the doctor, and the doctor ended up isolating this compound called phenylpyruvic. And basically, this is like a compound that's created in the metabolic pathway of breaking down phenylalanine, except that unfortunately, it wasn't able to be fully broken down because that enzyme lost its function. And so this is just an example of one enzyme losing its function due to a genetic disorder, and how the phenotype of that ends up being a whole range of things. Okay, so it also causes intellectual disability. There are other type of genetic disorders that affect proteins which act like hormones. And so dwarfism is actually a product of that. And it's basically a genetic mutation which codes for the protein that acts like a growth factor hormone. And so either there's less of the hormone being produced or more of the hormone being produced. And in this case, I believe it's less of the hormone being produced. Now let's go back to the hemoglobin example. There's a disease associated with that too, which is called sickle cell disease. But first, let's look at the hemoglobin protein itself. And so this hemoglobin here is in a quaternary structure. And if you remember, quaternary is a bunch of smaller proteins bundling up together to form a big protein. So this is a 4-protein structure making one big compound. And hemoglobin, we said that carries oxygen to all of the cells. And so it's a very important protein. And sickle cell anemia is a result of just one mutation, one nucleotide mutation. So it's thymine substituted for adenine. And so here you see T is the normal, and now A is in replacement of the T. And so you end up, as a result, you end up with a whole different amino acid in the sequence. And so instead of having glucine, that gets swapped out for valine. But yet this ends up being very detrimental. And the reason for that is because it affects the structure of the protein. It completely changes its structure. And so you notice here that this is a red blood cell that is normal. And so it has its normal protein structure, its normal 3-dimensional shape. And here, with just that one mutation, it completely changed the 3-dimensional shape of those proteins. And now you have this sickle cell looking protein. It kind of like twists the protein up like into like a tight rope. And so because of its loss in shape, it can't efficiently bind oxygen. And so as a result, there's less oxygen available to the cells, and then that gives a whole bunch of different types of phenotypic outcomes. And so some of those symptoms are weakness, abdominal pain, kidney failure, heart failure, just to name a few. And interestingly, sickle cell disease is, the disease itself is like there's like several types. And so if a person inherited 2 mutated genes, then they have the more severe type of sickle cell. But if they inherited just one mutated gene, it is still, that mutation is still expressed, but it's not the full blown sickle cell disease. And so are there treatments for the disorders that we just talked about, for the PKU, for sickle cell, and for other types? Well, there is some type of treatments, and you can go ahead and read about those in chapter 10. I'll leave that to you, especially since you have to fill it out in the study guide. So hopefully some of the concepts that I just reviewed helps you understand the chapter a little bit better. But I do really like the book. I feel like it's well written, and you're only responsible for 5 sections. So I think that, everything that I needed to cover has been covered. So just make sure you read the textbook, and also feel free to take a look at any of the YouTube videos that I have provided for you. The YouTube videos are just there to help you understand the information. Also, feel free to stop by my office hours if you have any questions or if you need additional help.